gesprochen.
What do you want?
No, it's okay, but I think it's going to be warmer as we... Okay, we'll put it a little later.
my great pleasure and honor to have Yuval, right, and that will talk with us about fMRI in the next today and next week. Yuval did his PhD with Uzari here, and now he's the director of the EMU, so the Health Tech Neuroimaging Unit. Whenever you want to think about MRI, and particularly of fMRI, he's the one that you should go and discuss it with. And we'll learn a lot from your knowledge about fMRI. Thank you.
My pleasure. Maybe we'll close another. Yeah, close the other one. So thank you for the introduction. No? But for me, it's good. I'm good now, at least for now. So I'm really going to talk to you, like Aviv said, about functional MRI. Okay. I'm going to focus today about the signal. We analyze the features of this signal, how we capture it, how we process it, and next week talk more about how we analyze it and different approaches people use. So just to start and get in the mood, usually see this kind of blob activation when you look at the fMRI images, right? This is one paper from 2010, and you usually what you think or what you inclined to think is, okay, these regions are active and in this context or the task that was given here, and you usually see all these bars that measure or represent the different activity levels. Okay, so that's a typical MRI, functional MRI result. And the idea, again, in this week and next week, is to give you some tools to learn to, first of all, understand how we get to these kind of figures, and to critically review them and understand what was done, or these kind of figures, they actually represent what they claim to represent. Okay. So we won't be able to do it all in 2 weeks, but at least give you some taste. So before we dive into the details, we have a long long and complex pipeline when we're talking about a functional MRI experiment. So of course we have some experimental design, some research questions that we want to answer. We have to translate that research question into something that we can test efficiently in the magnet. Then we acquire the data. There's some reconstruction, which all these stages I'm sure Aviv talked about or will talk about. Then there's a whole bunch of pre-processing algorithms and that we have to apply. And then we come to the analysis itself, which can be one of many different approaches to treat the data. Okay, and just to focus or concentrate on what we're going to talk about. So we're not going to talk about the data acquisition and the reconstruction at all, although it's very important. Okay, I'm here to talk to you about after you get the data, what do you do with it, how do you interpret it, and also considering that, how you want to design your experiment. Okay, because everything is connected. And in the analysis, we're also going to talk about the basically GLM and the classical analysis that you see, the contrast that you see, where something is more active. And although there are many more approaches that can be taken, we just don't have the time for that. So when we talk about design, we'll see some keywords like a block design, event-related design. All these designs you need to consider all kinds of parameters like the TR, the conditions you have, and so on. You have the pre-processing, which we'll go into in a second. And then in the analysis, again, just that this is kind of an intimidating slide. I thought about removing it, but then I said, okay, let's leave it intimidating a bit, or a bit messy, because there is a lot of lingo going around. And I just want you to know that you can do, when you come to to analyze the data, you can, first of all, there's selection of what you analyze exactly. So the raw data, or this is one option, and for that you can correlate the data, you can run a classifier, you can run some data-driven algorithm like ICA. do retinotopy, which is some kind of correlation. Also, you can, what you usually do and what we'll talk about is you can extract estimates from that data, and these beta estimates will serve as the hallmark of the activation, and those you can also analyze in a univariate way, which we'll talk about, or a multivariate way, which you probably heard about, or maybe I'll mention in a few minutes next week. And of course, you can analyze the whole brain or choose a region to analyze. So there's a lot of choices. Okay, you can some of the analysis done on single subjects, on multiple subjects. So there's a lot of choices you have to make and a lot of way to approach this. And like I said, everything is dependent on another thing, right? So when you want to design your experiment, you want to take into consideration your research question, of course, but also how you want to analyze the data in a way that will, enable you to answer that question. So, and also sometimes the pre-processing itself has to take into account the analysis or the research question. So, like I said, we'll talk about today the design and pre-processing, about the nature of the bold signal, and next week talk about analysis, and we'll focus about the classic univariate analysis, although I'm going to mention other type of analysis. Okay, so let's start from the beginning. It's always a good place to start. And this is what I'm showing you now is a very basic experiment. It's actually the first fMRI experiment that was done in 1992 in humans. And what you see here, again, it's the T1 map, it's the anatomical map. And you can see here, can you recognize this? Part of the occipital cortex, okay? And the cerebellum here. And without going into too many details, these guys made a really simple experiment. They flashed some checkerboard flickering at, I think, 10 hertz for several seconds. I think it was 10 or 12 seconds, and then shut the stimulus down, okay? And this was down in the right hemifield or left hemifield, so it will supposedly activate only one part of the brain, only one hemisphere. And what I'm going to show you now is activation timelines from 3 sites. Okay, there's 1, 2, 3 sites. This is the T2 star map. As you know, it has a lot. The resolution is much poorer than the T1. And this is what you see. Okay. So I don't think it's very complicated here, but you can already see difference between these two, right? So these two sites looks more similar, I would say, but in the timeline, and this one is different. And why is that, you think? First of all, what do we see here? Let me say. So here you have the onset of the light or the stimulus. Here it's gone. Here it's on again. And why would you think this? It's a very simple question, right? So why would these two follow this, the level of activation that you see here, follow the light, and this one don't? Right. Yeah, it's pretty cool, right? So you can see here. I'll show it more clearly. So this part is the cerebellum, more or less, okay? And this is the visual cortex, and these two sites are in the visual cortex, and it's not very surprising for us, but it was the first time that people show that intentionally, okay? So they designed the experiment like this. Shai, Mafgan, yeah, put on the air conditioning, it's pretty warm. So this is the first time they showed it, and again, they did some, took some measure to see that we're talking really about activation here, and they shown that if you show something on the, again, right hemifield, you see this pattern only in the left hemisphere, and so on. Okay, so it was a nice proof of concept that we can present something, shut it down, and we see the bold, okay, the oxygenation level rises and falls with the presentation of the stimulus. Okay, this is just a statistical map, and this brings me to the first key concept or a key idea. So when you talk about functional MRI, when you talk about any actually measurement that you take measure in science, you basically talk about SNR ratio, okay, signal to noise ratio. And in every paradigm, including fMRI, you want to make this ratio optimal for your need, okay? So really, in a very dumb way, just to ask you, so how would you improve the signal? There are two ways of improving the signal-to-noise ratio, basically. It's nothing to do with fMRI in general. Yeah, that's very, very simple. So, so you either improve the signal or reduce the noise or do something to both of them. And you can talk about signal and noise and in other terms, which are task-related variability and non-task-related variability. Okay, you have some variation in your signal, and you want to tease out the one that is relevant to your task, to your design. Okay, so but going back to what you said, can you think about now I'm really talking about MRI, ways to either elevate the signal or reduce the noise? We'll cover exactly what noise you have and signal you have, but I want to first hear ideas from you. So how can I improve the signal? How can I reduce the noise, control the noise? Any ideas?
Decrease sampling rate.
Sampling rate is one thing you could do.
Averaging.
Sorry?
Averaging.
What do you mean averaging?
Averaging across
Okay, so you have more data basically, more data points. You can average on those. That is a good way to do it. Another idea? How about removing the noise? Yeah, I didn't. Subtract? Subtract what? I don't hear. A baseline condition? What do you mean? Well, I can do something maybe related to that. Okay, if I have, let me help you with that. If I have some sources of noise, and noise here is everything that is not related to the task. Okay, I want to isolate the task-related variability. I could either first of all try to cancel the noise or cancel the noise sources, and the other thing I can do is, and this is something you might you were addressing to, is take the noise into account, okay? Be able to predict it, and if I have a good information about the noise, I can model it, okay? I can build a predictor for the noise, and then I can explain exactly what the noise contributes to the signal, even if I can't remove the noise. And about the signal noise, the signal itself, so everything you said is correct, but you can also do something in the MRI. You can have just a higher magnetic field. That also helps, although it also drives some kind of distortions and susceptibility. Artifacts are also more prominent. So it's a tricky business, but still, in general, when you do the same task in a 1.5 Tesla scanner or 3 Tesla or 7 Tesla, you get a higher SNR as you go up with the signal, with the magnetic field. All right, so we know what we need to do. So I'm going to spend the next few, or I don't know, 20, 30 minutes about talking about the noise and pre-processing and how and also the signal. And that's that. So what kind of noise can, maybe it was also in the paper, can you tell me what kind of noise are we expected to have in the fMRI environment? If you remember. Well, thermal noise, don't say thermal noise, it's on the board already.
Noise of the image when you resistant, like when you...
Right, that could have, yeah, you can have that. Head movements, all right, what else? You can have some kind of a regular frequency noise, like breathing, okay? It's not white noise, but it's a noise in context of your design, right? It's some kind of changes in the signal that you don't want to think that's part of your, basically a part of your design. So you're right. So we have some sources of noise that arrive from the magnet itself, so some thermal noise in conjunction with specific tissue of the tissue. It's not a nice word to say about a subject, because usually it's humans, but You have some interaction between your body, okay, and your tissue and the magnet noise, which you cannot predict exactly. You have some low frequency noise. You have changes, ongoing changes. If the task is like half an hour, you have some kind of linear trend and some really slow modulation that you want to account for. Then, like you said correctly, we have head motion, which is the most serious, I guess, problem. You have some cardiac, respiratory, and other circular or in some kind of frequency artifacts. And then you have some kind of variability that you cannot account for, okay? And these are fluctuations in the neural activity and, again, behavioral variability in the behavior and the neural substrate behind this behavior that are changing from person to person and along the, you have awareness and you have a lot of other things that you cannot really control for, okay? So you have some noise in the data, okay? But you want to, data is always noisy, but we want to remove as much noise as we can. Okay, so far we're clear? All right. So I'll go over these briefly. I don't want it to fill the whole day, but we do remove some of this noise. So again, like we said, we have this periodic noise, which is respiration, cardiac pulsation, among others, and the way you treat these are, a lot of people just ignore this, actually, okay? There is a way to basically account for these kind of periodic signals, and at least, for example, in our scanner, but in most scanners today, you have these measurement tools to measure your pulse and your respiration and so on, and you can actually build predictors, personal predictor for each and every person that is there, and you can remove this variability or this cyclic variability from the data. But so this is something we can track pretty easily, and if we think it might be in some kind of interfering with our signal, we can remove that if we want, or at least account for it. So the way to remove noise, or and also prepare our data for further analysis, is to do a bunch of processes that will basically, the idea is first of all, increase the SNR, increase the reliability of and the robustness of the signal, and also get individual data into some kind of normalized space that you can work on group analysis, okay? So we're going to go swiftly. Some we're really just going to mention, and some we'll spend some more minutes. And then, basically, you get, by the time you go over all these kind of processes and algorithms, you get some data that you can actually work with, okay? And this data that you work with, you then feed into one of your models that you have, or basically do whatever you want with it. Okay, so let's talk a bit about what's happening here, because I want you at least to be familiar with the term. So, slice time correction. Slice time correction is something that we have to do, and this is because of this illustration. So how do we scan a brain, right? You know about TRs, you know about TEs, you know about slices. So what's happening here? Can somebody explain me this? It's very simple, but I just want to know that we're on the same page. What's going on here? You have these different seconds, okay, running from 0 to 1.75, so it's a total of 2 seconds. So what's that? TR, right? So this is the time in which, the time unit in which we cover our whole selected brain. It could be the whole brain or just part of the brain, but this is our time frame, and here we have to work under this limitation. And what do we do in this time frame? We slice the brain, right? And we cover the whole box that we selected. All right? And we do it usually to escape some other artifacts that we can have. We do it in an interleaved fashion, usually. So this is scanned first, okay? And this is, you see by the seconds there, so we go 1, 3, 5, 7, and so on and so forth. But that creates a problem. What's the problem?
Measuring at exactly the same time.
Right. It's not exactly the same time, and in the end, you want 3D. You're looking at voxels, right? You want 3D image of your brain. and you want it to be unified at a certain time point. Your time resolution is 2 seconds. Every 2 seconds, you want one sample, and not usually it's around 40. Okay, 40 slices or 30 slices. So there are several ways to cope with this, but the basic thing you do, you have an idea. You read or you have an idea. You don't need to know it, but maybe somebody has an idea, or intuitively of what you would choose here, or what you would do. So what do we see here first? We see here slice 1, slice 2, slice 8, and again the slice is the location of the slice, right? So slice 1 and slice 2 and slice 8. So in temporal time, let's look at temporal time, the time, and not the ordinal number, right? We have these slices come first, and then number 3, and so on. And you can see that from the graphs, okay? Can you see the different onset, the different dynamics of the 3 slices? All right. So what would you do if you want, let's say, to get? You need to get one number in the end from that, right? You need to reduce this variability into one number. The idea. Sorry? Jittering. What do you mean? Yeah, you could do that, but that's a problem. That actually, yeah, that would be a problem because you're basically you want to stay time locked with your design, and we'll talk later about event-related design. You want to be pretty precise, but you can do something much more simple. You can average or take the time. I don't know if that's what you meant, but if not, then I saved you. But you just take the time of the more or less the middle slice. Okay, so you can do some other things. It's not the only thing you need to do, but this is something pretty simple that works pretty fine. Okay, but you should do that. This is just showing that, and I'm not going to talk more about this. Now, like somebody said here, one of the major things that disturbs our data is motion of the subject, motion of the head. Okay, this is can be very severe. So fortunately, there is 2 ways to deal with this, and the 2 are pretty robust way to do it. So the first is rule out moving subjects, or pick your subjects very well. If you have someone that already participated in MRI, this is why people pay a lot of money relatively to good subject in MRI. So if you want to make good money as a student, come early in your in the first year, prove that you cannot move in the MRI, then you get invitation to all MRI experiments, and you make some couple of thousand shekels during your depends how long you study.
But it's not allowed, right?
Really?
Motivation should never be money.
By Helsinki?
Should always be.
Yeah, you don't get. That's true. That's also true. That you're right. You shouldn't be. There's no benefit for the subject. But we pay everyone.
Okay, compensation of the time.
So
So for training.
true. So forget what I said. But I'm recorded, so I have a problem because everything is recorded. But all right, we'll talk about it later. Anyway, so that's one thing. But getting back into business, we have now pretty standard algorithms that take into account the motion. Basically, they sample the image you get at every time point, and they can detect changes in the intensities. Okay, and they correct for the head motion for most of the head motion. And the idea is that the assumption is that the head presumably moves only in rigid transformation. Okay, so it has 6 degrees of freedom, right? So it can move, right? There's a pitch, roll, and yaw movement to each direction on all 3 axes, and these are around the axis, and then you have the simple translation along the axis. Okay, so this is what the head does. Hopefully, nothing distorts during your MRI scan. So taking that into account, I don't know if this is a movie that works. Yeah, it works, but it's kind of hard to see, and it's very short and low resolution film. But you have something. You can look at this part, and these are this is the movie before the motion correction. This is after the motion correction. I don't know if it's very impressive, but you can see some kind. If you will really try hard, you can see some kind of rotation here and there. I'll show it one last time. Yeah, you have to see a lot of these, I think, to actually appreciate what you see here. But we can tackle this problem. Another thing you want to do is some temporal filtering. Now, I mentioned this before, okay? So, as you hopefully remember, there's some usually low frequency noise coming from the scanner, and we want to remove that. So just to, again, be on the same page, we can do high-pass filtering, low-pass filtering. Everybody's familiar with that? All right, so I don't need to talk about that. So usually we will do some kind of high-pass filtering to remove the very low frequency noise that we have, okay? Why we don't want to do so much of low-pass filtering? Again, intuitively, we can do some, but why we wouldn't want to do it as like just blindly, let's say? Where is our signal, the things we're looking for? Okay, maybe I'm not giving you the whole information. So I talk about really high pass, so removing the very low pass, very low frequency. I'm talking about really low frequency. So if you have a 5-minute scan, so I want to move something that is 2 cycles within the scan. So something very, very slow along 5 minutes. If I will move something, some noise or some filtering that will remove a signal that comes every 2 seconds or every 5 seconds or every 6 seconds, I just may remove actual data, right? Actual signal. I don't want to do that. So there are some situations in which you want to do it, but usually it's not part of the mainstream pipeline. So again, you have the data, which you see above there, and it's some kind of a composition between low frequency, high frequency, and the ideal activation that you have, the light on and off in this case. And this is an example of how a signal looks before pre-processing. These are TRs. So if it's 2, so it's 480 seconds. Okay, so 8 minutes. And you can see here some kind of modulation. I hope you can see. Okay, along the 8 minutes, and you want to remove that. And you want to remove motion correction and slice time correction, and you get something that is much more straight. That's the low pass, the high pass filter. But you can also see, even with the eye in this example, that you get some hopefully better SNR. Okay, so you get if we assume, again, I don't have the predictors, but if we assume that you have some kind of actual task-related variation here, it's cleaner. Everybody agrees with me on that? Okay. All right. We have time. By the way, you have a 5-minute break in the middle or not?
Could the filter interact with the experiment design?
Yeah, we just, okay. Yeah, it could then.
The filtering itself, I mean, when you here you try to remove bias because of motion, because if the motion goes with
right.
with your experiment or
Yes. So
experiment design.
yeah. So you usually don't want your two.
You might have very low.
Yeah. So first of all, yes. And you want to first be thoughtful about what you remove. So you usually remove at least automatically only the low signal, low frequencies. But also we'll get to that in the second half today about designing your blocks, events, or so on and so forth. For example, when you design a block, which we'll talk about in a few minutes, you want it to be not too long and not too short from different reasons. But one of the reasons you don't want it to be too long is you don't want it to covariate with some physiological measures or something like that. So, yeah. Hopefully, luckily, there's kind of a lot of evidence by now, and a lot of standard procedures that you can follow up with this. So, but do you have usually a break, a 5-minute break or not? Yeah, 15-minute break. Oh my God. Okay. So let me hold you for 5-10 more minutes. So we'll finish this part and then we'll go to the. I have time. Okay. You tell me when it's. If I don't get to my break, my planned break, you tell me. Do this. Suggestive movement. Okay. So. So right now we clean some noise from the signal, and now we'll talk about also some kind of noise removal, but also procedures that allow us to get into some kind of normal space or some kind of group analysis. So spatial smoothing. That's the first thing we'll talk about. And by that, again, without getting into the algorithm, we have the original data, which is today either 3 by 3 millimeter voxels or 2 by 2, even 1 by 1 if you have a strong enough magnet. You get this voxel, you have an intensity for each voxel, and you might want to smooth this by averaging some kind of averaging with neighboring voxels, okay? Create a smoother picture. Now, why would you want to do that, you think? I already kind of hinted at that. What do you get by basically you have larger units, right? Now, so did some interpolation, you have larger units. What's the pros, let's say? What do you gain? What do you lose? But maybe first, what do you gain? Bigger voxels, and what does that give you?
It's less sensitive for motion, for movement.
It is less sensitive for movement. It's also what we talked about before. Sorry?
You have a better signal.
Right. You have so when you're talking about a lot more surface, basically more neurons, you have more signal, more data. You have some more reliable activity. That's very good. That's one thing. And the other thing that I just started mentioning, maybe not clear enough, but okay, let me help you. So you have, like you said, motion artifacts, but you have also variability in our basically brain, and also variability in the way we measure different subjects. So if I want to compare one voxel between two subjects, the activity of two subjects, or average, and so on, if I have a larger unit, I'm more able to do that, okay? Because I just can, right? So if I have a very, very high resolution, then matching voxel to voxel will be harder, okay? So that is a very good a reason to do spatial smoothing. Why shouldn't I do spatial smoothing? Yes, but how?
Two voxels that have different signals, now taking just one signal.
Yeah, you lose your resolution basically. That's true. And even to more extreme, you can use some voxels that are significant can become insignificant, or the other way around. It's really what you said. But it's taking one voxel that wasn't active, okay, and the other one that was active, and then you combine them, and what you're left is some average activation that is not really significant, okay? That's one example. And you can actually lose some clusters. If you have very small clusters, right, that let's say 2 voxels that are important but are surrounded by less active voxels, so averaging all this out, you get something very non-significant. You lose basically that cluster. So this is just how this kind of data looks, okay? So you smooth again and again. Let's see if we skipped something. So we talked about SNR and increased the overlap between subjects. And we talked about lower resolution, mergers, separate activation loci. So not only you can lose activation loci, but we can separate, join or merge, separate activation loci. Last thing, almost last thing you want to do is co-registration. So, as you know by now, you should know, you have T1 images, that's your anatomy. You want you have your data, functional data, which is in different resolution, taking either a different session within your day of scanning, okay, with a different protocol. Use EPI, not MPRH. So everything is different, and you have to have, in the end, you want to talk about where in the brain something happened, okay? And you want to compare between your research and previous research, and you want to compare between subjects. And for that, you have to co-register, basically, your functional data with the anatomical data. And again, luckily today, we have very, very successful algorithm that detect these kind of landmarks and intensity changes in the MRI and in the anatomical maps and the T2 star maps, and they basically do this kind of, again, transformation to put everything in its right place, okay? And there's a bunch of files, of header files that connect your two types of data. But last but not least, you want to normalize sometimes you want to normalize the space in which you work. And what do I mean by that? Right now I have the data, I pre-process everything, remove the motion artifacts, I put it on the brain of that subject, but I'm still in native space. What does it mean? It means that when the subject, the patient was in the scanner, the head was somewhere in the middle basically, and there was some kind of, we put this square, right, this box which we scan. This box has some X, Y, Z coordinations, but these X, Y, Z coordinations are not exactly the same as a different subject, okay? Because the head is not positioned, it's exactly the same space, and the sizes are different of the head and the brain and so on. So in order for me to talk about a voxel that is in, let's say, X, Y, Z position in space, and compare it to different work or different subject, I want to put everything together in one normalized space, and for that I need to do some kind of transformation on the space for each of the subjects, okay? And basically there are two these kinds of transformation, rotation and translation we talked about before with the motion correction. Here we do also scaling, okay? So we do some scaling of the transformation, and rotation won't help us because the brains are just different, the sizes are different also. And just to, this is what you assumably would get, right? You have these different locations and sizes of the brain, and you get something unified. And just to, again, put you with the lingo, we have two ways of doing this. One is Talrach space, which is, has its cons and pros, but it's basically a one brain on a very old woman that was measured, when? 20 years ago. Talrach, the original.
Separate.
No, but measured.
You mean the Brugman one? The one woman brain, or the Talrach that they
project? Yeah, the one that I don't remember. In the 90s, I guess, no? 80s. Okay. No, it doesn't matter. But basically, there's, you have a template of the brain, okay? You have landmarks. You recognize the tip of the nose and the ears, stuff like that, and then you align the brain, the specific brain, magnify it, and rotate it, and so on, to put it on the same template. Another approach is the MNI space, and I'm just mentioning it because it's a different approach. This is based on a morphed average of hundreds of brain. It keeps growing also, this database. So you have some kind of, and it looks morphed, okay? It looks more, when you look at it, I don't know if I have, no, I don't have it here, but it looks kind of a morphed brain, more average brain, okay? And why choose one of, there's a reason I don't want to go into now, but basically, in the end, what happens is some softwares, okay, for all this processing, we use some software. This could be SPM or FSL or Brain Voyager. I don't know if you heard about it. But every software uses one of these approaches, this template. So in the end, what happens is usually you go with your software. But I think, from my experience, I think I would like to go with MNI, even though I did Talrach by now. But the idea is, again, you want to have a normalized space so you can do some group measurement and relate your work to other works, okay? And I think this will be the last slide for this part of the talk. The last thing you do in this pipeline is you don't have to do it. Usually you can do analysis in the volume space. Okay, these are the matrix, the bit matrix of voxels that you basically collected, the data you collected, and you can take the gray matter here. Again, some by now standard algorithm that inflate this brain. They paint the algorithm paints the sulci in dark gray and the gyra in bright gray. Okay, and you have this inflated brain, which especially for visualization, but also for some type of analysis, is better. Okay, but this again, we don't have time for that. But I just wanted to know that this is the final step you do on a need-to-do basis, basically. All right. For me, this would be a good time to have a. All right. So we move to analysis next. What's that? Ah, okay. וואלה. I'm not going to talk too much about it just because I don't think I'll have enough time. We didn't talk about yeah, maybe in the end, because I want to talk more about more the functional part of the data itself, and less about decisions. I think you got a lot of that. But you can ask, we can talk about it if you want. Yeah, I'll be happy to.
נכון, אבל כאילו שווה להעביר מסרים. לא, אנחנו שקלים שאתה... הכל חשוב. הכל צריך לקחת בחשבון. אבל אני חושבת שיש לו משהו מפחיד. כאילו, ממש התחלתי כזה... לא, אבל זה כל זה. נכון, לא, אני מסכימה, אבל... כן. אבל זה לא הגיוני. הוא לא... שזה תמיד שאתה עושה את הרוב. זה הגיוני. כן, עכשיו 8 שמענו, כמו שאמרת. ומה זה 0.6? זה 600 מיליסקנד. 0.6 שניות, בסקנד. לא יודע מה עובד, זה נכון. זה שם נכונים. לא, 0.6 זה לא שניות, אם זה 600 מיליסקנד. זה? 1000 שניות. אבל אולי נכתוב במיליסקנד. לא. למה? ופה זה אגב חייב להיות מיליסקנד. כן, פה אפליקציה. וזה מה? זה כולם, זה הכל. כן. לא, זה... אז סבבה, אז מה שהוא רוצה זה שנעשה את זה רק ככה. כאילו את החלק הזה, ניקח רק בפיק הזה. כן. ונעשה את הממוצע כמו שאמרת, נכון? כן, ממוצע על כל...
time. Dat is wat de referentie. To by się wyobraź fajnie. Nie, to nie jest tam jest fajnie. Ale to ja napiszę to tych kolegów.
So I was asking a question. Where's the shop? Right? Right on the top of the possibility.
Tak?
Not doing the things now.
Right, when I come in, I'm done. See? Like, correspondence, email. But people actually answer to email. Can I use your paradigm? Something weird about it.
All right.
They're happy to everybody.
Okay, everybody's okay? Caffeine level back to normal? Okay? Right. I want to switch gears into the analysis part. Well, not exactly the analysis, more the design part. But we'll talk about how it's connected to analysis today and talk more about analysis approaches next week. So let me first introduce the main, the lingo, the main design paradigms that people use. And we'll go over and understand the logic behind each and every one of them, and also understand some other things about the volt signal, some other features or the nature of the volt signal. So the most simple design is the block design. This is something that you saw earlier. It's very simple. I have a condition, switch the condition on, whatever it is. Let's say a light on. I wait a few seconds, okay? In that period of time, I initiate some kind of, let's say, a visual stimulus, okay? So it could be an image of a face, let's say, or several images of faces, okay? I finish with the block. I give some time to basically rest. Why would I do that? Very simple. Sorry? That?
So you can return to baseline?
Baseline, the volt is baseline. Another thing, that's the main thing, but they're also behavioral, okay? Behaviorally, I want to create some separation. But you're right, that the long period of rest is because I want the ball to return back to baseline. And so this is the basic thing I want to, if I want to have a very, well, not only simple question, but something that I can capture with a basic, simple design. Okay, I don't have to have 2 blocks, type of blocks, I can have 7 types of blocks and so on. What do you think are the main advantages, disadvantages of such a design? Again, we'll dive into that, but out of like top of your head.
Easy to analyze?
Sorry?
It's easier to analyze?
It's easier to analyze, easier to interpret, and to actually to prepare. That's true. If I show a repeating stimulus, let's say 20 faces, one after another, would that give me a robust signal or no? What do you think? Yeah, you can guess. It's like, okay. It will give us, right? Yeah, you're right. So it's a very robust signal. I show 10 seconds of consecutive stimuli or 20 seconds of consecutive stimuli. I get a very robust noise. What am I'm missing something here. I'm not missing, but something is not very efficient here. What is that?
With expectation?
True, one of that's a behavioral consideration, right? If I have some kind, if my behavioral paradigm, a research question, demands that I introduce a stimuli or a condition in an unexpected way somehow, that's a no-go for this one. But in terms, another like basic limitation, I always kind of a lot of time here. Okay? What I will do, and we'll show you next week when we talk about. Oh, I need to open this. All right. So now we're online. What we'll do next week, we'll talk about GLM. We see that we build this kind of predictors that collect the data from the epochs of activation. Okay. So if I have a block design, I typically will have several repetitions of each blocks. Why? Because each block takes 15 seconds, and then I have like 15 seconds of rest for the ball to go back to baseline. Then I have another block, and staying of more than a few minutes in one session or one run in the MRI is problematic. Okay, it's problematic behaviorally. People get bored, fall asleep, and so on. So I'm not very efficient in time here, but I do get a nice robust activation. I could alternatively divide my data into events, single events. Again, if here there was a lot of faces, here I show one face, then I wait, then I show one house, let's say, then face again, house, and I randomize this. This is called event-related design. And the first one of the two is called slow one because I wait for the signal to go back to baseline. Okay? It will enable us to do this behavioral manipulation that we weren't able to do before, okay, like excitation, stuff like that. It will have significantly less statistical power, or I would have to have a lot of repetitions like this to get the same statistical power. And it, as you might guess, it's very, very non-efficient in time in the temporal domain. So I really am wasting a lot of time here, and time is also not convenient or not comfortable for the patient or a person inside, but it's also money. Okay, we're very constrained. Experimental MRIs is kind of expensive stuff. You want to minimize the time in the MRI. So I think we'll see that in a couple of minutes again, but I think it's clear the disadvantages here, but there are some advantages here also, and I'll get back to that in a few slides. Then we have a rapid jittered event-related design, which is a version of this design. And again, we'll talk about it in a few minutes, but you can, talking about these two, you can understand why this design is, well, it is efficient, and it is, there's a lot of reasons why you want to use this design. There are also some limitations, but one question you should ask yourself is how is this possible to analyze, assuming that, or what we know of the ball, that is? The delay, exactly. The dynamics of the ball, right? So it takes a few seconds to go up, then it depends on the stimulus, you have some plateau or not, and then quite considerable time goes down. How do we basically get or tease out the real data if we repeatedly induce a stimuli without waiting for the signal to go down? And this is something people dealt with, and you can do it. And then we have some mixed models, which you have some blocks and alternating events within a block, but we won't touch that. It's just another like a small deviation of the typically used designs. So when we talk about our design and then our analysis of the fMRI signal, we talked about a convolution that we make with the onsets, okay, of the different stimuli, presumably driving neuronal activity, okay? And we have to convolve it with, or multiply it with the HRF form, which again, I'm sure you talked about it with Aviv. They have some kind of canonical form. There's a lot of variability, but when building, and this is important, when we build some kind of model or we have some assumptions on our signal, we assume we have this canonical shape, and we assume that all voxels and in all voxels and on all conditions, this shape stays the same. It's not always true, but we have to assume that to account for the other variability, okay? And the idea, again, if you convolve, so if you have this hypothetical input vector right? You have a short or weak positive input and then a strong positive and then negative and positive. If you multiply each one of these components with the HRF, you get this set of multiplication, compound signal, and then you put them on a timeline, and you get some kind of signal. Of course, there's noise. You have to add noise. But that's the main idea. And it turns out that there's a very clear relationship between the supposed neuronal activity, or let's stick with the temporal onset of the stimuli. Okay, when we talk about design, let's not talk about neurons. And if we have some kind of a predictor that describes the neuronal activity or the onset of the stimulus, a short stimulus, then a longer one, and so on and so forth, we get this boxcar predictor. We put it together with the canonical HRF, and we get expected response. Okay, it's very, very simple. And for the block design, and now I'm going to dive a bit into each one of the three designs I talked about, we get this typical plateau here, right? So we have this initial dip. You talked about that, right? You talked about the ball, the social ball, right? You have some initial dip. You have the rise. Again, this is scalable to a certain amount by the stimulus intensity, stimulus length, okay? But you have some kind of rise. You have basically saturation. You cannot keep rising. But then if some stimulus continues to go on, you get this long response, and then the decay after you finish presenting the stimuli. Okay, so in a block design, this is what you see, more or less. So we're good until here, right? This is pretty simple. And let's take a single block experiment. Again, very simple, light on and light off. You have this very, very clear predictor here, okay? In this part, the light is off, light on, off again. And what you, if you just convolve this with the HRF, you get this simulated signal here, okay? So you have the timeline on the X, and this is second, but it can be in TR. Now, if you have some kind of rapidly alternating neuronal activity or tasks, changes in the condition of the task, this is what you get. And why is that? Again, you talked about it, so just don't be afraid to say it again. Right. There's no time for the signal to basically return to baseline. Okay. But that's okay. Now it depends what we want to measure. If we want to measure the initial rise in the signal, we're good. If we want to measure different events here, because this block is different from this block and so on, we're not good, right? We have a very, very low energy, okay? We have a very, very, relative to the changes that we could have here, the signal, if you want to check the difference between the blocks, is very small, okay? This is the signal in this case. This is basically the signal here. So we don't want to do that, okay? So that's one thing. And of course, everything is more complicated or even like less clear because we have noise. So first of all, we have noise in the MRI signal. We talked about this noise, okay? So it can add some kind of linear trend, but also some kind of high frequency noise. And then, if we're talking about neurons in this case, we have to remember that the neurons, we have some embedded noise in the neuronal activity and therefore in the ball activity also. So in adding this noise and this noise, in the case of long blocks with long baseline periods here, we'll get something noisy, but we get a lot of signal here. And if we use the other example, of course, we left with basically, in this specific simulation, more noise, okay, so random noise, than the actual signal, which is periodic. So this is just to illustrate that you have to think about how you design your experiment. and specifically for block designs, is that they have their, again, it's very simple, it's very straightforward. You have a lot of detection power. Or if you want a contrast between two things, it might be very good to use it. But the blocks shouldn't be too short. Then you basically lose your whole detection power. And they shouldn't be also too long, by the way, because, like we said before, they might be removed very, very low high-pass filters. So low signal, if it's very, very low, like every 1 minute you have a block, you'll probably filter that out. Okay? So this 16 seconds is something that it can be like 12 seconds or 10 seconds or 18 seconds, something like that. And then, of course, we have the rest periods. Okay, so this is very simple, very good for certain kind of questions, less so for other kinds of questions. So next in line is the slow event related design. So again, the idea is the same. We have this onsets, offsets, okay? We have the expected modeled HR response, and we put them together. We get this kind of expected MRI signal, and as you can see here, you have a lot of signal here, okay? If you use this slow event related design, you can see, assuming that this voxel, okay, this is a simulation, but assuming the voxel is actively responsive for that specific stimulus or that specific task, okay, you get a very nice signal. Of course, if a voxel is irrelevant to the task, you'll probably see something flat there. However, like I said before, let's see if you remember, what's the main problem with this kind of design? It's a long time, yeah. It's very boring for the subject. It's you get less data basically, okay? But it has its advantages. And one of the advantages is a very high estimation power. So when you talk about signal in general, and now we talk about bold, basically you want to do two things, either detect changes or estimate let's say the time course, the dynamics of the signal, okay? Or maybe do both. Usually when we talk about MRI, when you hear about MRI, it's detection, right? I want to see the level of activation in this condition and that condition, see if there's a significant difference, okay, and go and start working from there. If I want to estimate, that is, see how the stimulus, intensity, length, whatever, affects the shape of the HRF, okay, then I want this HRF to be time-locked with a single stimulus, and I want to see the whole evolvement of the function, basically, of the ball, all right? So if this is my purpose, then I want to see something that's event, just one event, and gone, and then I want to give the ball time to evolve, okay? So this is when I would use this kind of design, the slow event related design. So again, it allows the estimation of the HRF. I'm not gonna talk about this, but some kind of, when you want to assess motion artifacts, it's also beneficial because you can estimate the response. If you want to, let's say, tease apart some elevated response, okay, you have some elevated response, and you want to see if it's a motion artifact or not. If you have the whole timeline of the response, a motion artifact behaves differently, okay, than an HRF. All right. When you have let's say a block design, okay, and the task creates motion artifacts. So I hope that you see this slide, but if this is the block and you have motion artifacts, so you have some motion artifacts along the block, because you don't see the actual response function for every stimuli here, okay, you see the whole thing, so it's very hard to tease out the motion artifacts from the stimuli. But if you have just this reaction to one stimulus, and then let's say motion artifacts, because someone speaks, let's say, the motion artifacts will look differently. If this is the time point of where the trial starts, the motion artifact probably will be something like this, okay? So it's very intuitively, it's a different response, and when you have good estimation, you can tease between these two. like I said, we have a poor amount of data, and it could be very boring and tedious for the participant. Are we good until here? All right. So these are the two main things that people use in the beginning, and then people thought, okay, we need to have a better way to do these things, and we want to introduce some, first of all, events that come more rapidly, and introduce also a different level of expectation and jittering between the different conditions. And of course, if you, it's not only that you get more data when you do things rapidly, you can think of different designs, okay? If you need some designs that, again, you need to present something rapidly and then an audible experiment, audible condition that surprises you, like you said, I think. So this is a very good design. But to do this, we have first to prove or to show that the bold can be deconvoluted, basically, into separate components, okay? And how these components interact. So if I have one bold response that starts here, and then before it goes away, there's another bold response and so on. I need to be able that I can reverse engineer the overall signal into the components. And I can do that if these components are, interact linearly, okay? And there are several experiments that show this. I'm going to show you one of them, done in, I think, 97, one of the first ones. The stimulus here was very simple. Again, a checkerboard. You fixate here in the center of the screen. There's a checkerboard flickering, and this checkerboard flickers for 1 second, if I'm not mistaken, maybe 2. I think 1 second. And they had several conditions, okay? The first one is that you have this 1 second stimulus, and then there's rest, and then there's another one. The second condition was pairs of stimuli, okay? 1 second stimulus, then 5 second delay, and then another second of stimulus, okay? So that's a pair of stimulus, and then they have also a triad of stimuli, okay? So 1 stimulus, 2 seconds baseline, then another stimulus, 2 second baseline, and so on. But then after this triad or pair or single trial, there was like 15 seconds of rest for the signal to come back to baseline, and they tested activation in visual cortex, and this is what they saw. So what you see on the left, in one subject, you see the response or the activation for one trial. Okay? This is when I have 1 second of stimuli, and then when I have 1 second stimulus, 5 second baseline, and another 1 second stimulus, I have this, right? So the response climbs here, starting to drop, but then goes another one. And they did a very simple thing. They did it repeatedly, but the idea was very simple. They subtracted one from another, okay? So they subtracted this from this, and they estimated the pure response for the second stimulus, okay? And this is what they got. So the first trial is this, and the second trial, the estimated second trial is very, very close to that one, okay? So this was the first indication that there is some kind of linear summation, okay, of the response to each stimuli. They later presented this. They switched this timeline or this activation into basically one value per voxel. Okay, they reduced the data into one value per voxel, and here you can see the distribution of the activity in the visual cortex, and you, without again going into details, you can see it's strikingly more or less the same. Okay, and again, this is one trial activation, and this is the you see 2 trial clusters with 1 trial cluster subtracted out. So this is the estimated second trial activation. So it's pretty much the same as the first trial, the real activation, the real, well, measured, not estimated. They then went on to do this with 3 trials, and let me save you the details. And as you probably would have expected by now, they saw that this also continues when you add more and more trials. Okay, so again, estimating the second from subtracting the, let's say, the all trials, you can subtract 2 trials and get the third one, and so on. They did all these kind of checks and tests. And you can see that the first trial is basically have the same shape, the same magnitude of the second, the estimated third trial, the estimated second trial. So the bottom line here is we have linearity of the bold. Okay, this is very important. 1 second. This is very important because again we have some kind of tool that we have some way to model the bold, right? Because we know where exactly we put the stimuli. And we can infer even if we have some overlap between the conditions. Yeah?
Why there is no undershoot for the first trial and just for the estimated second result?
Good question. Let me think about what they did. Let's go first for the first trial that you see. Let me see if I can see it clearly here. No, it's always together. It's a good question. It may have something to do with the baseline, with a normalized baseline. I'm not sure. I can maybe look into it and then see for next week. It may have something with the way, because I guess they estimated from baseline to the next baseline. Right. Yeah, well, change from baseline, right? Yeah. So I'm not sure how they related to the baseline. I just don't know, right? It seems like, yeah, the estimated, actually, you would think that the estimated signal here is it's behaving more like the canonical HRF than the actual measured signal. So yeah, I'm not sure. Probably something about the baseline, but I can check. All right. So this allows us, again, we don't have time to, it's a whole course doing that. By the way, there is a course about this. I'm teaching this year and probably in 2 years, because I don't think we'll do it every year, that goes over all of this and all of, especially in the visual domain, and talks about MVP and stuff like that. So you can learn more about it. But unfortunately, I cannot go into that now. So I just want to tell you and trust me that, at least get the intuition that you can treat the different parts of the bold. You can linearly combine them, linearly sum them, okay? And this gives you the flexibility to use this very rapid experiment design. And of course, the advantages is that it's less boring, that enables, like we said, oddball experiment, enable the estimation of the HRF to a certain point, less so than the slow event-related design. Again, we won't go into that. And another thing that it does is saving time, of course. That's very, very important. Yeah, I think that's enough for that. Okay, so, but there's another thing. This is clear, more or less? You get the intuition for that is clear, I hope. Okay, so there's another thing I want to talk about. That's the jittering of the rapid event-related, and not only rapid event-related trials. And what do I mean by jittering? We talked about it a bit before. We're jittering in the sense, usually we do it in the scale of the TRs, and we want to create some offsets between the expected, or between the expected frequency of trials. Okay, and why is that? We want to introduce more energy to the system. Okay, and I said now the bottom line. Let me go back and retrace what I said and explain what I mean. So in the very simple case, when you have two conditions. Oh, I'm missing a slide here, I think. Oh, that's too bad. Okay, so the slide I'm missing here is what you have when you don't jitter. Okay, and basically, I hope that you see what I painted, like wrote here before. Is if you don't see, tell me. I'll turn on the lights. But basically, when you have one condition that goes on, and then another condition go off, and another go on, and another go off, in a very systematic way, very periodic way, what you usually get is some kind of anti-correlation between these conditions. So every time this condition is up, the other one is down. Okay? Even if you take into account the changes, the dynamics of the HRF, okay, of the function, the canonical HRF is the same. Okay? So it's when you give it in let's say this condition 1, and here condition 2, condition 1, condition 2 again, 1, 2, 1, 2, 1, 2, right? And it's very periodic, so you have some kind of rise. Let's do it like this. You have some kind of rise here, let's say, and then for condition 2, let's say you have less activation, and then another point, condition 1 and condition 2, 1 and 2. I didn't draw it quite well because there should be more overlap, but you see it here. So if you had like one of these and one of those, one of these and one of those, you had some, again, anti-correlation, and what you get in total signal will be something more like the short blocks we had before. Okay? So we get some kind of rise in the beginning, of course, and then you get some kind of this little fluctuations between the two. Okay, so every time you get some a constant combination of the two factors. Okay, one time right now is the peak of the first condition, and the second condition. Let's say this is the first and this is the second. So here you have the peak of the first. Here you have the peak of the second with some kind of leftovers from the first condition. Here you have the peak of the or here you have the peak of the first again, and some leftovers from the second condition, and it's very periodically. So you get two states basically all the time. Okay, again, intuitively. What you want to do is introduce a jitter. First of all, randomization in the order, and also a jitter. You move the onset of specific trials a bit forward or backwards, okay? And then you create more variation in the data. So if you have 3 consecutive yellow conditions, you get some kind of different behavior of the total amount of signal, and then you have a couple of purple ones. You have a different combination, different total, different summation. Again, because it's all summated linearly. So you have a different summation, and if you move them in time a bit, you get more variation. And again, you can do that because later you can tease these apart, right? Because you know that you have some kind of response function. It's part of your model. You have the onset, okay? So you can deconstruct this overall signal, okay? So this is what enables us to do this. And again, the name of the game here is to have as much signal as we have, so as much variation and noise that we have. And this is kind of complicated to do. It's very hard to do, and usually people do 2 things. Okay? Stay with me. It's not it's like a few questions. I think that you're getting tired. This is kind of a technical, boring thing, but it's important. And I'm finishing it in 2 minutes. So what you usually do is one of 2 strategies. Either you have a lot of data and you randomize everything, okay? You just randomize the test, the trials, and over a lot of data, you get a lot of variability, and basically you have a very high signal. Alternatively, there are some algorithms that optimize. Okay, you give the conditions that you have, you give the length of the condition, you give the HRF function, and you give amount of basically blank periods that you want to put, like units of blank periods, and this algorithm optimizes. They optimize the sequence, the specific sequence you need to place these conditions in order to achieve maximum energy. But for me, I just want you to have the intuition, okay, of why you should do that. Okay, this is when you introduce jitter again. So this one introduce random. This is 2 types with random. Just random assignment, but this is the next step, which is jitter, and here you get an even more signal. Okay, more changes in the signal again because you introduce this asynchronous synchronous onsets. All right, so let's sum this up. This part. So when we get the next thing we'll be talking about analysis, we always have to remember that our design, we want to make our design in a way that will match the analysis we want to do, and the analysis we want to do is based on the question of a research question, basically. Okay. So when we consider the design, we first of all need to think what the person needs to do, and what feature, what aspect of the signal are we looking for? Okay, are we looking just to detect changes? Are we looking for some kind of estimation of the signal? The bold response itself? Are we just looking at group statistics and so on? In general, large variance mean large signal. Okay, so like we saw, block design with very short blocks is bad. Of course, in a block design, real block design, you only look at the beginning of the change. So you can do that, but you cannot relate to specific trials within the block. Okay. When the ISI, so interstimulus interval, isn't fixed, so we add jitter, rapid sequences can achieve very high efficiency. And to sum up, block designs are great for detection, poor for estimating the response itself. Slow event-related design are poor detection because we have less data, but they're great for estimate. And fast event-related designs have basically good detection, also pretty good estimation. And again, it all depends on your choice of research question. Okay, so this is it for strictly design issues. From here, I'm maybe start just introducing analysis, and we'll do this next week. But first, I want to, if you have questions or if something is not understood from what I said now. Again, I don't want you to remember like numbers or specific details, but I want you to have the intuition of what we did every stage of the pre-processing, what kind of designs, basic designs we have, what are the pros and cons, the consideration. Okay, so I think, yes,
And about the summation.
Right.
And so isn't there saturation of the block?
Yes, there is. There is saturation.
So that is
Yes, there is saturation because there's just an amount of blood you can pump into a certain voxel. Okay, if you just show, I don't know, 100 stimuli after another, you won't put like 100 liters of blood there, right? So you have some kind of saturation, but the length is also modeled. So if I have what I showed you before, the summation, so they did 1, 2, 3, okay? They did, let's say, 1, 2, 3 trials, and they have a couple of seconds difference between them, okay? If they had like 15 or even less, I'm talking about, I don't know, 5, 6 trials with no difference or very small difference between them, they won't be climbing, and at a certain point they will be saturated, and only the length will change. But you can model that. That's no problem. And again, you won't usually get into that because you introduce some jitter and some difference between the events. Okay, so it's true that if you have an efficient design, so you should have some kind of variation that will get you a bit drop in the signal before you elevate it. Okay, so you don't want to go into this plateau where you reach in block design when you did summation in the beginning and then you just stay there. That's not efficient. Okay, but if you design it correctly, you won't get there. You can generate it, you're right. That answer a question or yeah? Okay. More questions? Okay. So that's good. Let's see if we have like 5 more minutes. So I don't have everything ready also for next week. This is what I wanted to talk today, but I can at least introduce what we're going to talk about. So let me just give you the framework of what we're going to talk about next week and repeat what I said in the beginning. So there are many types of analysis approaches that you can take when you're doing fMRI, and again, this is kind of an introduction, like method introduction course. So I just want to have this 3 minutes to put you in the lingo of things. So one of the basic decisions you have to make is, are you working on the raw time course, basically, right? So raw time course, it means that you have basically x, y, z, and t. So for x, each voxel, which is represented by x, y, and z, right? You have a vector, right, of activation, timeline of activation, and this is t, right? This is time. So you have a big matrix, x, y, z, and t, okay? One of the, what people usually do is try to have some assumption, and these assumptions are derived from your design, okay? So if your design says light on, light off, you have some assumption that visual cortex voxels will show more activity with lights on, less activity with lights off. This assumption is implemented with your design, okay? So you have some expectation of how this region, how this voxel will behave, okay? You can, and we'll talk about next week, you put this assumption into a model, right? And then you extract some estimates. And for now, we just need to know that these estimates reduce this timeline. Okay, so if I have, these markers are horrible. If I have, and I'll finish with that. If I have a timeline for a voxel, yeah, I can take again, I can deconvolve this. I understand that in this time point I presented a face, and in this time point I presented a house, and here I presented the face again. Okay, so I can take the component of this timeline. I estimate the component that belongs to face, that was contributed by by showing the face, and I take all repetitions of this component and reduce it to one value, basically. These are my beta, my estimate. So these are the first distinction that I do. I can work on the time course on the betas, and then if I'm working on the betas, and this is what we'll talk about, work about the estimate, we can analyze each and every voxel separately, or we can analyze the pattern, let's say a vector of 200 voxels, and see the combined activity. This is the multi-order variant. But we'll talk about it next week. So unless there are many more questions, I think we're good. Yeah? Okay. See you next week. חביתם את המזגן עכשיו, נכון? איך חם. חום.
זה בעיה, ההפתעה הזאת ממש בעיה.
כן, זה בינארי גם.
כן.
זה הבעיה. מה הלוגיקה של זה בסדר כאילו מבחינת להיכנס לדברים שאני אחיד יותר משהו כבד לפחות כן, ללכלך על אנשים. לא, בדעתי זה היה בהתחלה, בקצה ראשון אני יותר קצת. כן, אבל זה אמר לי זה מה שהוא טוען, אולי הקורס הזה לא היה בדיוק שלו שעשה. כן, יש כמה כוכבים אבל, אני חושב שאתה מורה, כאילו, טובה, כי יש מישהו, למרות שזה... כן, אתה סלקשן בייס, כן, אתה כולל את האלה שמודים. קיצור, לא, בסדר, חוץ מהסוף. Oh yeah? Oh no, I didn't remember today. I didn't remember today. מה? תקרא, זה לוקח לו זמן. לא, צריך לעשות לו אוף זה, אני יודע, אני נבחר, אני לא נשמע אותו, אבל צריך לעשות את זה. נתראה שבוע הבא.
ולא בדרך
תבואו אליי.
מה קורה? חזרת מהציוד? היא הפחדן הזה, כתבתי עכשיו, ממש באו אנשים פה שאתם התעניינו וזה, דברים שלך, כן, קראת פה את שזה וזה, מתי הוא חוזר? תחילת יולי? ראש סתם, באמת, רגע אי אפשר בכלל, זה גם און-אוף, זה רק און-אוף, ופה אתה אפילו כרגע לא יכול להדליק את זה מתוך כמו פה. כן, זהו, כן, היינו צריכים ללכת לשלטר כל הזמן, להגיע להוא שילך החוצה, להדליק את האור. קיצור, יאללה, יורד למטה.
En pratique, c'est sûr que je veux dire, si tu prends un homme n'importe lequel dans la rue, il y a des chances que... Il y a des chances. C'est la norme qu'il est...
Tu le vois souvent même à l'aéroport, en Tunisie ou ailleurs, souvent avec la femme.
Ah oui.
L'heure.
On attend la réponse. J'étais relancé là et il répond pas. Et même David répond pas, j'ai envoyé un mail et là. Toi aussi t'as fait le coup David récemment? Ouais. Parce que là, il était il était en partance. Là il est aux États-Unis. Ouais. Et il était il partait le 4. Ouais. Donc moi je lui ai envoyé un mail le 4, je me suis dit si j'ai pas de réponse, je lui ai dit colloque, mais si j'ai pas de réponse c'est normal, il y a un avion, décalage horaire, bonne, c'est même normal si 2 jours après j'ai pas de réponse. Mais au-delà, ça devient inquiétant. Ouais, là on va voir en fait Gaël, là j'ai commencé à taper on va le voir là j'étais en train de taper le compte rendu, un début de compte rendu écrit qu'on va rendre, et comme ça on va pouvoir lui présenter ça déjà. Apparemment, le conseil Charard tient les portes parce que son suivi encadré et puis il n'avait pas du tout fait ce qu'il fallait, donc j'espère que lui c'est pas mal, il y avait un peu, c'est assez varié en fait, parce qu'il y a à la fois du spectral, Ah, okay. So no
It's
worries.
just your computer. Still. Why is that? But maybe the version? למה זה בקאפ version יש לך?
נשארת פה שעה בינתיים?
כי זה לא עובד אצלי ולא
אולי
אצלם.
אבירם ושירלי יקדימו ואת נעשה סדר.
ננסה נשב עם
למה?
נשב ולתת להם זכויותיכם.
כן.
אתה ראיתי את הסגנון הזה.
So אליהו אמור
ההורים שלך לעשות בדרך? מגיעים גם?
מה?
ההורים שלך מגיעים?
כן.
זה היום?
אין לי כוח בכלל. תדע תחפש משם.
זה גם מרגיש לי כאילו, מילא אם הייתי מקבלת משהו...
כן, כאילו כבר למדע המחשב.
כן, או תואר שני נגיד.
כן. ונראה לי שגם כשאתה מגיע לתואר שני אתה כבר עובר אותו דבר על דוקטורט.
כן?
כן, לפני שאמרת תואר ראשון אמרת וואי, עשיתי תואר ראשון בחיים אני משוגע.
באמת?
כאילו, זה כבר עכשיו שיאללה, אני עם תואר. כאילו, זה...
אבל נגיד התואר הזה חסר משמעות, זהו. האמת שהשלב שהבנתי את זה זה שאני אמרתי, כזה היה טקס תואר שני לאח שלי השבוע, וסבתא שלי הייתה אמרה ללכת, ואז אמרתי לה, אני גם מקבלת תואר השבוע, והיא אמרה, אבל זה בטח המונית כזה התואר ראשון. היא אפילו לא חשבה על לבוא.
You can go if you want, I will join you in...
אבל באמת כאילו אין שום סיבה שהיא תבוא, כאילו, אבל זה גם...
כן, לא, יש לה ספלים לעשות, כאילו, היא יכולה כל הזמן להתכתב.
מתן! כל כך הרבה צרות אתה עושה.
זה לא טוב.
זה לא טוב לך. נהיה טוב.
טוב, יש לך בנוגע לתהליך.
I just can't unplug it because there's no battery, so I have to... I can't unplug it because there's no battery, so I have to switch it off properly, but here it's blocked. So if you want to go, you can just go and I will join you. I can even try to send you the... maybe I can force...
Maybe keep it the document is in the
זה יצא שהוא לא טעים?